I'd like to thank the organizers for inviting me here to give a presentation. I work in the areas of plant oils, trying to understand how plant oils are formed and also modifying the plant oil formation process for the benefit of mankind, looking at both food and non-food applications. And we have a network of researchers that we interact with. We have various funding agencies that support our research efforts. I wanted to bring to your attention two major programs we're involved in. The Bioactive Oils program has been running for a few years now, and it's funded by AVAC, and partners are involved in that. And one of the main goals there is to develop trans-free solutions for oils for the baking industry, and also to develop polyunsaturated enhanced flax for health and animal applications. More recently, the Phytola Innovates Center was funded, and that extends some of the work in the bioactive oils program, but also the major goal here is to increase oil content in plants. And there are other sub-projects there as well. And the Phytola and the Bioactive Oils program are interested in, of course, developing interactions with industry. And we sort of look at ourselves as the early phase of this pipeline leading to a product. So I'll talk about seed oil, some major oil crops, talk about canola, and then give a couple examples of the research we're involved in, oil content work, and then talk about some of our work on developing designer oils that have anti-cancer properties. And I'll talk about our general goals and the team expertise that we have. Oil essentially is a triacyl glycerol, and the nature of the fatty acid chain that's connected to the glycerol backbone determines the properties of this oil. And so if you have a lot of double bonds here, the oil will be more liquid at room temperature, whereas if the double bonds aren't there, it's going to be hard like lard. Interestingly, if you look at these so-called hydrocarbon chains, they're essentially the same thing as you'd find in petrochemical products, for example, octane being a component of gasoline. And so not only can we use oils for applications in food, such as vegetable oil, but we can also, or as I say, something you put on your salad, but you can also use oils as precursors for biofuel, as well as various production of various chemicals that are similar to those derived from petrochemicals. These are the major oil crops in the world, and we can see where we stand. Our major oilseed crop is under this oilseed rape umbrella, and canola is under that group. And that accounts for about global production 2009-2010, about 22 million metric tons, and worldwide oil production is about 140 million metric tons. And the leader being, of course, palm, and another major player, soybean. So a lot of competition here in terms of the sorts of oil profiles or fatty acid compositions that are being generated, and to use those oils for various feedstock applications. Canola, it's obviously important in Canada. This was a major scientific milestone development of this, and it's helped us quite a bit. Oil content is very important. As time goes on, we're going to need more and more oil, both for food and non-food applications. So it's very important to have research that focuses on increasing the oil content of seed. There's also other research going on where they're trying to convert, trying to get vegetative tissue to accumulate oil as well as in roots. So it's coming from all directions, even algae, a lot of interest in getting algal lipids formed. So it's anticipated that there's going to be up to 75% increase in oil canola production required within the next several years. Interesting, a 1% increase on an absolute basis in seed oil content in the canola seed will result in additional $90 million per year, and that benefits the crushing and processing industry. At the present time, producers don't get extra money for growing high oil content lines as they do in Europe. Oil formation occurs during seed development. You've all seen the canola field. These flowers give way to a developing silique or pod, and if you crack it open, you see the developing seeds with the ovule, the seed coat on the outside. If you peel that away, you'll find this embryo forms from heart shape to late cotyledonary stage. And within the oil-forming cells of this tissue, the oil will accumulate. And here's where all the biochemistry goes on. And the biotechnologists are interested in modifying those pathways. So this is a very simplified representation of the oil formation process, or at least part of it. The enzyme I've worked on since the late 80s is diacylglycerol isotransferase, or DGAT, and I always like to say one day I should put this on my license plate. And this process essentially involves the addition of fatty acid chains to a scaffold with glycerol as a backbone. And eventually, the very final addition of the fatty acid chain is catalyzed by this enzyme to form the oil. And it's been shown that this enzyme, there's many plants, including canola, this represents a bottleneck in the oil formation process. There's a lot of biochemical evidence for that. And if you can somehow open up that bottleneck, you could potentially make more oil. And there's been experiments, and we've been involved in that, where you get more of this enzyme, and we in fact get more carbon deposited in the form of the triacyl glycerol. This is some data from our group, and also there's been some work by Monsanto and other groups. And what we've done here in canola is we've taken a naturally occurring form of the enzyme that's already present in the seed and just got the seed to make more of it. And so we've demonstrated under greenhouse and field conditions that we can get an increase in oil content. Also, Calgene Monsanto, they've taken a fungal DGAT and expressed it in soybean. And so they have a product in the pipeline where they have a higher oil content soybean. And it's also being looked at in corn as well recently. What's interesting too, this is my colleague Dave Taylor from the Plant Biotech Institute as part of our team. He had grown a variety of canola, and it had drought conditions that year, and oil content was severely depressed. So the control was giving a value, it should be in over 40%, was closer to 30. But the transgenic lines, which were overexpressing the DGAT, there was a reduction in the penalty on seed oil content caused by drought. So it's sort of a side benefit. So build in some insurance into the seed against adverse environmental conditions in terms of penalty on the product you're trying to develop. So we still have to investigate that more. It's kind of interesting, though. More recently, our group has developed some technology focuses on directed evolution. We're trying to take the DGAT enzyme and trying to make it into a better performer. And so we do this through a random mutation process, and then we use a screening system to select for more powerful enzyme forms. And this technology is under, we have a patent application for this, and it was, publications appeared in the same year. And so this is essentially the work of, mostly the work of Dr. Rodrigo Silotto in my lab as a research associate. And through this technology, he has identified one very active form here, and you can see the increased amount of triacylglycerol formed in that case. And it's just showing you the more active form. We also work on flax, and that's a very important crop as well. We're collaborating with a nutrition research group, Catherine Field, to look at development of polyunsaturated fatty acids in flax. We have nutritional data to demonstrate, for example, in flax, there is a lot of alpha-linolenic acid present in these breast cancer cell line. These regular sorts of fatty acids don't seem to affect the growth of these cells, but a number of polyunsaturated fatty acids with extra double bonds will in fact inhibit the growth of these cells. And so we're interested in further developing that. And these are our goals in research and commercialization in our group are to, of course, increase oil content, increase saturates for certain applications, flax oil for aquafeed, nutrition, high-performance lubricants. Can our expertise be used to develop other oil-based products for industry? Phytola is seeking additional industrial partners. We are currently engaging a number of industries and are almost closing a deal with one major group. This is our team expertise. I just flashed this up here. We have everything from analytical chemistry, genomics, computational biology, right through business development covered. Nancy Cranston's in our audience now. She's the business development director with our group. And this is acknowledge a number of interactors, team players, both within the university and outside. And further acknowledgements as to funding sources. And finally, I just want to mention that we are having an oil conference in Edmonton, and if you're interested, to mark your calendars for September 26th. There'll be a dinner at the Devonian Gardens along with this, and this is the full spectrum of oils: nutrition, processing, engineering, genetics, everything. And we're going to mix everything up, so the nutritionists can't vanish, but everybody's going to be there at the same time. Thank you.